Hey everyone. In the last video, you learned about some of the genes that regulate the cell cycle. Now, in the next two videos, we'll take a look at the basics of cancer genetics and what happens when those cell cycle regulation genes become mutated. As always, remember to fill out the learning guide at the end of these videos, and by the end of watching both of them, you should be able to answer 3 questions. And our 3 questions are: how does cancer develop? What are the characteristics of cancer cells? And how does mutation of tumor suppressor genes and proto-oncogenes lead to cancer? There's a lot to these questions, so we'll take 2 videos to answer them. Before we get to it, I'll offer a quick disclaimer here. I am a doctor, but not the kind that you need if you want answers about your individual health or cancer risk. I have a PhD, not a medical degree, which means my training is in science, not medicine. Science is finding out how the universe works, or for me, how genes work. Medicine is getting people healthy and keeping them that way. Very different jobs that require fairly different skill sets. So any questions you have that pertain to your own health or the health of others should be directed to your medical provider. And nothing in any of my videos should be considered medical advice. Cancer occurs when cells divide in an unregulated way. Cells dividing in this way, over time, can eventually form a tumor, which is perhaps the most well-known aspect of cancer and is what many cancer screening methods look for. In this pair of videos, we'll look at what causes this uncontrolled cell division and, therefore, what causes cancer. In the last video, you saw how the regulation of the cell cycle prevents uncontrolled cell division. If cell cycle regulation is lost, uncontrolled cell division results. Other processes are also important. For example, if apoptosis is lost, this can also contribute to uncontrolled cell division because cells that have lost control of their cell cycle are allowed to survive and keep reproducing. Loss of DNA repair can also contribute to the uncontrolled division of cells because DNA damage that causes loss of cell cycle regulation is not repaired. For some cancers, all 3 of these things must be lost in order for uncontrolled cell division to occur. Loss of these essential processes is caused by the loss of the proteins that carry them out, which happens when the genes encoding these proteins are mutated. For example, the nucleotide A at a given position in the gene could be changed to a G, and this might destroy the function of the cell cycle regulation protein. Different factors can cause mutations, including chemical compounds called carcinogens, which literally means cancer-causing substance. Some viruses can cause mutations because they can insert themselves into or near the DNA of genes and change their expression. Nuclear radiation from radioactive elements and UV radiation from the sun also cause mutations. However, most mutations are caused by naturally occurring errors in DNA replication, because the enzyme that carries out DNA replication is not perfect. When DNA is replicated, each strand is separated and used as a template for the synthesis of the new strand. But if the wrong nucleotide is placed in one of the new strands, a mutation occurs. So what causes cancer, ultimately, is anything that can cause a mutation. If the mutations occur in genes responsible for cell cycle regulation, DNA repair, and other processes like apoptosis, uncontrolled cell division and eventually tumor formation can occur. This is basically how cancer develops. And note that most mutations do not result in cancerous growth. It's only those mutations that mutate genes that actually control these processes that can actually lead to cancer. Now, mutations come in many types, and I'll talk about a few of them here. A substitution mutation occurs when one nucleotide is substituted for another. This is the most common type of mutation. An insertion mutation is when one or more nucleotides is inserted between two existing nucleotides, making the DNA sequence longer. This G was inserted between these two T's. In deletion mutations, one or more nucleotides are lost, making the sequence shorter. Insertion and deletion mutations in a gene are more likely to destroy protein function because they're more likely to drastically change the amino acid sequence of a protein. Fourth, a DNA sequence can be duplicated. Occasionally, an entire gene can be duplicated. If that gene's function is to promote mitosis, the additional copies of that gene will result in increased amounts of protein, which can cause cell division rates to be higher than they should be. In other words, uncontrolled cell division. The increase in gene copy number by duplication is called gene amplification. A fifth type of mutation is chromosome translocation. Now, before translocation, all genes on these chromosomes encoded distinct proteins. During translocation, the DNA of each chromosome is broken and reattached to another chromosome. If the break point occurs within a gene, that gene might no longer encode a functional protein, and that would cause loss of cell cycle control. Or, as shown here, if the break occurs in a way that puts one gene right next to another gene, this could result in a gene fusion. These genes might be expressed as one single gene and encode what we call a fusion protein, which has parts of both proteins that each gene encodes. Some fusion proteins can cause uncontrolled cell division. A translocation between chromosome 9 and chromosome 22, as shown here, is responsible for a form of leukemia called chronic myelogenous leukemia. Any of these mutations have the potential, if they occur in a gene that regulates cell division, to cause cancer. Recall that the cell cycle has several checkpoints. If a mutation causes one of these checkpoints to not work, in many cases, the other checkpoints will still prevent inappropriate cell division. For many cancers to develop, multiple checkpoints must be bypassed before uncontrolled cell division can occur. This usually requires multiple mutations to at least a few different genes. This is one reason many cancers require several mutations and the loss of multiple checkpoints before they can occur. Let's take a look at how this might happen. Here is a somatic or body cell. Let's say it divides by mitosis, as somatic cells like to do. But a mutation occurs in one of the two daughter cells. And it occurs in a gene that regulates the cell cycle and cell division. This single mutation isn't enough to cause uncontrolled cell division most of the time, so we don't see cancerous growth yet. The unmutated cell continues normal division, but when the mutated cell divides, it will pass on its mutation to both of its daughter cells. Perhaps a second mutation occurs in this cell. Once that cell divides, it passes on both of those mutations to its daughter cells. Now a third mutation arises and is passed on. So by chance, some cells accumulate more mutations than others. Now a fourth mutation arises. And in this example, it's this mutation that finally destroys enough checkpoints and cell cycle regulation is lost, resulting in uncontrolled cell division. Once the cell starts dividing uncontrollably, it is said to have undergone transformation and is now considered a cancer cell. Perhaps a fifth mutation, here, increases the rate of cell division even further, causing this 5-mutation cell to outcompete the 4-mutation cells. This is known as clonal selection. Remember that clones are genetically identical cells. These cells, with the same 5 mutations, are clones of each other. The 5-mutation clone is selected for because it reproduces more quickly. This should remind you of natural selection because that's what it is. Natural selection can be an important part of cancer development and the growth of tumors, which is what this group of cells is becoming. One cell of this tumor might gain an additional mutation and pass it on to its daughter cells. That gives them the ability to detach from other cells and move to a different part of the body and reproduce there, forming a second tumor. This is called metastasis. Cancers that metastasize become much more dangerous because these secondary tumors can form in other organs, interfering with their function and threatening the life of the patient. Now, mutations don't occur as often as I'm showing here. There could be many cell divisions that occur between this first mutation and the second mutation. Perhaps this first mutation occurred in a stem cell when this individual was just an embryo, while this one occurred at age 20, and maybe these two occurred at age 70. The more time passes, the more mutations can occur. This is one reason cancer is more common in older people. They've had longer for more mutations to accumulate. More cell divisions means more chances for mistakes in DNA replication. Different types of cancers require different numbers of mutations to develop. On average, it's 11 for colorectal cancers, 6 for lung cancer, 4 for breast cancer, 3 for ovarian cancer, and 2 for retinoblastoma, an eye cancer that we'll discuss in detail later. The mutations we discussed on the last page were sporadic, meaning they occurred within an individual's lifetime. But mutations can also be hereditary, meaning the mutation was inherited from a person's parents. Of all the mutations that drive cancer development, most are generally sporadic, while fewer are inherited. A mutation can be inherited from either the mother or the father, through either the egg or the sperm. If either gamete carries a mutation, and here it's the egg, during fertilization, the zygote will inherit that mutation. Since all cells of an organism are derived from the zygote via mitosis, all cells of the embryo, and eventually the individual, will have that mutation. Usually, one mutation is not enough to cause cancer, but the existence of one mutation in all cells means that one less mutation is needed for cancer development. And this increases the chances of cancer occurring. For example, a woman who inherits a mutation in the BRCA2 gene, which functions in DNA repair, has about a 60% chance of developing breast cancer by age 70. For those without this mutation, the number is only 12%. Because the inherited BRCA2 mutation is present in all cells, this puts these women at increased risk of other cancers as well. This is why doctors routinely test women with family histories of breast cancer for mutations in BRCA2 and other genes. If someone has a family history of breast cancer, one of their parents is more likely to carry the mutation and could have passed it on to them. This testing is important because early detection leads to better patient outcomes. It's also important to realize that someone with a heritable mutation can pass the mutation on to their own children, because the mutation is present in all of their cells. If the mutated gene is still heterozygous, when the gametes develop, only half of their gametes will have the mutation. Sporadic mutations are not passed on by parents, so the zygote doesn't have any. However, when the zygote divides by mitosis, mutations can occur in one of the somatic cells. This can occur at any time, early in embryonic development, as shown here, or very late in life. If it occurs early, a patch of cells derived from the mutated cell will also carry that mutation. Just as with heritable cancer, it will take more mutations for sporadic cancer to develop, if it ever does. Sporadic mutations are not passed on to offspring because somatic cells don't pass on their DNA to gametes. Okay, let's take a break here. I'm going to enjoy a beer, and if you can, I suggest you enjoy a beverage as well. See you in the next video.